Okay, so let's start with chapter 1. Okay, so chapter 1 is going to cover on genetics and genetic inheritance and genetic control. Okay, so for chapter genetics, because it's going to cover for 3 weeks, so there will be a lot of information that we are going to, facts that we are going to be looking at. So for the notes, there will be two parts, okay, part 1 and also part 2. Okay, so part ada dua PowerPoint lah, nanti ada dua file, okay, yang you all kena download nanti. Kalau dalam new future ni, dia dah keluar. Ada dua lah, ada part 1 and also part 2, okay. So we are going to look at part 2 first, because we are going to learn about DNA and also protein synthesis that we are going to apply during the lab session next week. Okay, so these are the content for chapter 1. Okay, so we are going to look at some terms in genetics. You have to know genetics terms such as genotype, locus, allele. Okay, phenotype, homozygous, heterozygous, and all. So all that terms you have to know, you have to understand. And then, so Mendelian genetics, monohybrid inheritance, dihybrid inheritance, extension of Mendelian genetics, human pedigree, and then after that, population genetics, terms in population genetics, Hardy-Weinberg equation, the Hardy-Weinberg equilibrium. For this one, there will be a bit of calculation that you have to do. Tapi tak susah pun, dia tak sampai nak kena ada derivative semua, tak ada. Just simple equation that you have to apply, okay. So this is for the part 1. Okay, so this is part 1 of your notes, and then part 2, and so part 2 is this one here. This is the component for part 2, the subtopic. So for part 2, we are going to look at the DNA structure and replications. The structure of DNA double helix, we have looked at DNA structure, RNA structure last semester during chapter 1, macromolecule. So I expected to you to be familiar already with the structure of DNA and also RNA. Okay, the models of DNA replication, and then Michelson and Stolz experiment, DNA replications, and then after that we are going to proceed on protein synthesis, which is involves these two processes, transcription and also translation. So types of RNA, basic principles of transcription, translation, genetic code, transcription of RNA, modification of pre-mRNA in eukaryotes, translation, which is building a polypeptide, and then regulation of gene expression, component of an operon, lactose operon, and then we are going to look at mutations, small-scale mutations, chromosome mutation, human disorder due to chromosomal alteration. So this covers on the second part. So, but we are going to look at second part first, because we are going to be applying the knowledge during the experiment. Okay. So basically for the chapter genetics, okay, the base, our cells, it is composed of DNA. Okay, so DNA stores all your genetic information. So from that DNA, the information will be used to synthesize protein. Okay, so protein. So that large information of DNA in your nucleus have to copy a small section of that information into a smaller molecule. We call it as RNAs. Okay, so there's 3 types of RNA. tRNA, mRNA, and also rRNA. Ni pun kita dah tengok last semester kan. tRNA is transfer RNA, mRNA is messenger RNA, rRNA is ribosomal RNA, component of ribosome. As we know, ribosome is the organelle that involves in protein synthesis. Okay, so from the information in the DNA, copy into a smaller molecule RNA. That molecule, it carries information to the cytoplasm. It goes to the ribosome for protein synthesis. Okay, protein synthesis. So protein synthesis, the monomer is amino acids that will be assembled into a polypeptide chain. So this protein, polypeptide, becomes a protein which have many functions. So from the protein, it is used to make enzyme, it is used to make hormones, it is used to make other proteins that the cell requires. Even your muscle is made up of proteins. Okay. So and then this one gene expression sama lah, daripada information daripada DNA pergi ke RNA sampai ke hasilnya protein tersebut. But this one we are going to look at how gene are regulated by a group of genes. We call it as like operon. So usually this gene like operon are usually found in bacteria. Okay, like operon. So the bacteria will be using the lactose to metabolize the lactose as the source of energy. So how does the bacteria uses that lactose to synthesize, to be used to be used as source of energy? It is controlled by like the lactoperon. Okay. So and then mutations, right? Mana rewriting? Okay. Okay. So this is learning outcome dia tak ada. Tak apalah. Katanya ada learning outcome Oh, learning outcome dia kat sini. Sekejap. Learning outcome dia kat sini, sorry. Sebab dia kat part 1. Okay, so these are the learning outcomes, the learning objectives. So I explain the importance term in genetics, describe Mendel's principle, law of segregation and law of independent assortment, explain examples of extended Mendelian genetics, describe human pedigree analysis, define terms in population genetics, describe the Hardy-Weinberg principles, explain the factors that change frequency in a population. So kat sini kita akan banyak tengok on crossing. Phenotype, genotype, crossing, dapat anak dia apa genotype, phenotype anak tersebut. Okay. So that one, this one we are going to be focusing on the inheritance of specific characters from the parents to the progeny, from the parents to the offspring. Okay, so there will be some calculation of the frequency of the inheritance of specific genotype or phenotype. Okay, so there will be some calculation based on two formulas that we are going to look at. And then describe in brief the structure of DNA double helix. This one we have touched during last semester, chapter 1. And then cite evidence from Michelson and Salt's experiment regarding DNA replications. So during mitosis and also meiosis, DNA will be replicated. So how does the DNA replication occurs? We are going to look at in this part. DNA replication also can also describe DNA replication, synthesis of leading and lagging strands, what is leading strand, what is lagging strands, how these strands are synthesized, explain the nature of genetic codes. So the component of DNA and also RNA, it has bases kan. Kita tengok ada adenine, guanine, cytosine, thymines. Okay, so for one amino acid to be coded by the DNA or RNA, it must consist of 3 bases. We call it as codon. Okay, each amino acid has its own codon, has its own genetic code. Okay, so 3 bases is the codon that codes for specific amino acids. That will lead to the synthesis of polypeptide to become certain proteins. Okay, explain the process of protein synthesis. Explain the concept of operon and gene regulations. Describe the mechanism of like operon in the presence and absence of lactose. So this one we are going to look at a group of genes that is involved in the metabolism of lactose in bacteria. Okay, so and then define mutations and then describe the types of mutations. Okay. So these are some of the genetic terms that we are going to look at. Ini kita cover nantilah sebab ini part 1. Okay. So saya nak pergi terus kepada part 2. Okay. DNA structure and replications. Okay. So DNA structure and replications, DNA double helix, Michelson and Salt's experiment, and also DNA replication. Okay, so basically the first subtopic here it says DNA double helix, the structure of a DNA strand, So DNA molecule is a double-stranded molecule, okay, double-stranded. So this is one of the strand, okay. So as you can see, the monomers for DNA is deoxyribonucleic acid, okay. So the monomers for DNA are nucleotides. Okay, so nucleotides. So this is the structure of one nucleotide. So a nucleotide is composed of a phosphate bonded to a 5-carbon sugar bonded to a base. Okay, so there's 4 types of bases that are found in DNA, which is thymine, adenine, guanine, cytosine, and also guanine. Okay, so the bases are grouped based on its structure. So whether it is single ring or double rings. Okay. So if this DNA is double stranded, so thymine will pair with adenine on the other strand. Okay, so a double strand. So in thymine will pair with adenine will pair with thymine, cytosine with guanine with cytosine on the second strand here. Okay. So another point that you have to take note on the structure of DNA is that it has directionality. Okay, so because this is a DNA molecule, the 5-carbon sugar, the pentose sugar, we call it as deoxyribose. If this is the monomer, the nucleotide for RNA, the 5-carbon sugar is ribose. Ingat balik kalau RNA, the 5-carbon sugar is ribose, kalau DNA is deoxyribose. Apa beza ribose dengan deoxyribose? Is at carbon number 2. Deoxyribose, carbon number 2 has hydrogen. For ribose in RNA, it has OH. Okay, OH here. And then RNA, the difference is that instead of having thymine in RNA, it is replaced with uracil. Okay, so the same structure. So and then the DNA strand, okay, the one DNA strand, it is consists of the sugar phosphate backbone. Okay, so the sugar phosphate backbone is consists of a repeating unit of phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. And then for each strand of DNA, it has directionality. Where to find the directionality? When you are looking at the structure of the 5-carbon sugar, so 5-carbon sugar at each point here, it has carbon, right? Okay, so it is a pentose sugar. So this is carbon number 1, carbon number 2, carbon number 3, number 4, number 5. Saya tukarkan dia pada pen. Okay, so this is one nucleotide. Okay, so one nucleotide. So carbon number 1, 2, 3, 4, 5. The carbon number 5 of one nucleotide, it is bonded to its phosphate. Okay, so therefore the DNA strand starts with a 5' end. Ini permulaan pada DNA strand. Okay, it starts with a 5' end and ends with a 3' end. Okay, so 3' end, as you can see, this the 5-carbon sugar where it has the third carbon where it has originally a hydroxy group, it is where condensation reaction occur to attach one monomer to another monomer. Condensation reaction to attach monomers together. So through the removal of water molecule actually. Okay. So condensation reaction occurs to attach monomers together, to attach the nucleotides together to form a polynucleotide. Okay, a polynucleotide. Ingat balik ni chapter 1. So condensation occurs attaching the nucleotides together. So whenever you are going to add new nucleotide to the strand, you always add at the 3' end. Okay, so because there's OH here, so the hydroxyl will be removed and there will be one hydrogen from the phosphate also will be removed to remove a water molecule to attach the next monomer from the previous monomer, the next nucleotide from the previous nucleotides. Okay, at the end of the DNA strand, so it has a free 3' carbon. Ujung dia akan ada 3' carbon. So if there will be addition of another nucleotide, the next nucleotide will be added at the 3' end. So the DNA strand will start with the 5' end, where it is attached with its own phosphate, and at the end it has a 3' end, where it has a hydroxy group ready to be added with the next nucleotide. Okay. So and then another thing that you have to take note on the structure of a polynucleotide is that each nucleotides are attached through the formation of phosphodiester linkage. Okay, so phosphodiester linkage is the bond between adjacent nucleotides. Okay, so there will be possibility of questions. I'm not sure whether it is asked during semester 1 or 2, but you have to know the facts. Where is the location of the phosphodiester linkage? It is between carbon number 3 and carbon number 5 here. So this is the phosphodiester bond. Okay, between carbon number 3 phosphate and carbon number 5 of the next nucleotide. So this is the phosphodiester linkage. Yeah, phosphodiester linkage here. It attached between adjacent nucleotides. Okay. So the sugar phosphate backbone is the alternating phosphate and sugar molecules from which the bases projects. And then another thing that you have to take note, why does the molecule call it as DNA? DNA, deoxyribonucleic acids. Okay, due to the property of the sugar phosphate backbone, it is negatively charged. As you notice, it has negative charge. A negative charge where originally it releases hydrogen ions into the solution. Okay, so the property of a DNA, it is negatively charged. Okay, negatively charged. Okay, and then ini penting untuk awak tahu. This is really important for you to know because the knowledge on the facts that DNA is negatively charged will be applied during chapter 2. So ada application yang kita akan belajar nanti. Dia akan tunjuk migration of DNA in a certain apparatus. So the movement of DNA will be from positive to negative. From positive to negative. Macam tu. Okay, so negative to positive. Macam tu lah. Okay, so kenapa movement dia macam tu? Sebab DNA is negatively charged. Okay. So and then the directionality of a polynucleotide strands is from 5' end, okay, with a phosphate group, and to the 5' end with the OH group of the sugar, hydroxyl group. OH is hydroxyl group. Okay. Okay, so next, the structure of DNA, we know that it is double helix. Okay, so this model was proposed by these two scientists, Watson and Crick, model of DNA. So the double helix model of a DNA molecule was discovered by these two scientists in 1953. So the two strands of DNA form a right-handed double helix, right-handed. Okay, so dia pusing daripada kanan ke kiri, right-handed. Okay, so the two DNA strands are stabilized by hydrogen bonding between the base pair. So this is one DNA strand. So this is the second DNA strand. Okay. So as you can see, the DNA strand for the first strand here that starts with the 5', the first nucleotide, it has the base thymine. So the pairing with thymine is adenine. So to pair thymine with adenine, the attachment is through the formation of hydrogen bonds. As you can see, every time when there is hydrogen bond, it is always drawn with a dotted line, with dotted lines. Okay. So as you can see from the diagram here, the formation of hydrogen bonds between thymine and adenine, there's 2 hydrogen bonds. Between guanine and cytosine, there will be 3 hydrogen bonds. Okay, 3 hydrogen bonds between guanine and cytosine. Adenine and thymine is 2 hydrogen bonds. There will be 2 hydrogen bonds. Okay, so the basis pairs is according to the Chargaff's rule. Okay, so Chargaff's rule basically it says that adenine has to pair with thymine, guanine with cytosine. So and then as you notice, there's the 2 DNA strand, they are running in opposite direction, anti-parallel. Okay, so the two polynucleotide strands are anti-parallel in opposite direction with regards to their 5' to 3' directionality. Okay, 5 to 3. So kalau one strand, kalau saya nak draw kat sini, dia start dengan 5, akhir dengan 3. Arrow dia ke sini 3. Kenapa arrow? Sebab ada OH kan. So kalau DNA tu nak dipanjangkan lagi, if you were to add new nucleotides, always added at the 3' end kan. So 3'. So the complementary DNA strand here, macam tu kan. The direction is 5 to 3. Okay. 5 has to pair with the 3'. 5 has to pair with the 3'. So 5 ke 3. Sini ada OH lah. Okay. Okay, so they are running in opposite direction. The two strands are running in opposite direction. Okay. So I'm going to play a video. I'm going to play a video. Okay, so basically this video, it explains to you on the structure of DNA in comparison with RNA. And so DNA is double helix, double-stranded, and RNA is single-stranded. Okay, but when we are going to look at later the structure of tRNA, for example, all RNAs are single-stranded. Okay, but for the structure of tRNA, because there's the structure of tRNA will be folded into specific shape. The folding of that tRNA causes the bases to also pair up. When the bases pair up, doesn't mean that the RNA consists of double strand, but it is due to the folding structure of the tRNA. The RNA is folded into specific shape. We call it as clover leaf shape, but RNA is single-stranded. Okay, single strand straight line. Okay, that will go to the ribosome for protein synthesis. So in the last part of the video, it mentions that the DNA it has certain sequence of nucleotides. Okay, so the nucleotides are arranged into specific sequence that the some region we call it as genes that code for the synthesis of specific protein. So specific protein will have certain sequences of nucleotides to produce certain sequences of amino acid to form specific protein. Okay, so dia tak boleh suka hati je susun A, G, T, C. Dia ada susunan dia yang mana dia akan menghasilkan satu kod untuk menghasilkan protein. Okay, so the coding of the DNA will be copied in the RNA so that the RNA can synthesize protein. If you look at this diagram here,
DNA and R.
okay, so this is 3 bases kan. So setiap 3 base we call it as codon. Okay, codon, okay. 3 consists of 3 bases. Okay, so 3 bases. Macam tu every 3 bases. Macam tu. For example, the base sequence AGC will code for certain amino acid. Okay, so there will be specific amino acids to form specific protein. So this protein can be enzyme, can be hormones, can be other proteins that the cell requires. Okay. Okay, so from DNA to chromosome. So as we know, the monomer for DNA is nucleotides. Okay, so that are attached together, assembled together to form polynucleotides. Two strands of polynucleotide forms a DNA molecule, double-stranded helical structure. So because our cell is really small, that alone the nucleus is really small in a cell. So this is a cell. Inside there's the nucleus. Kita tengok kita punya kulit ni, kita tak nampak pun satu sel tu kan. It is really small. The cell is really small. You have to observe under microscope or buat eksperimen sel dulu kan. So nampak bawah microscope. So in the nucleus, there will be the DNA. Okay, so there will be the DNA. This DNA has to be organized in a certain manner. Okay, so in order for the DNA to be arranged properly, neatly inside the nucleus, as you can see, there will be protein. We call it as histones. Okay, globular protein, we call it as histone, where the DNA strand will coil around the DNA, the histone protein. So from DNA coil around histones to form chromatin. Okay, to form chromatin that will be condensed to form a chromosome. So this is the structure of chromosome. Okay, so as you can see, a chromosome, it has very long strands of DNA. Itu satu chromosome. Yeah, so satu chromosome. Kalau manusia, we have 46 chromosomes, where we have 23 pairs of chromosome to form 46 chromosome. Okay, so 23 pair to have 46 chromosome in a cell. Okay, so just imagine berapa panjang DNA dalam satu sel dalam badan kita ni. Okay, macam itulah. Okay, so this is the size of the chromatid. So really small. Okay, so the molecular structure of DNA double helix, the Chargaff's rule. Okay, so this is the Chargaff's rule. So DNA base composition varies between species. Okay, so the percentage of adenine and thymine bases are roughly equal as those of guanine and also cytosine. Ni buat ni guanine and cytosine bases. So this rule implies that base sequences within the two DNA strands are complementary to each other. So the first strand, for example, so this is the sequence, okay? So the second strand which runs in the opposite direction, anti-parallel tadi, kan? Anti-parallel. So 5 has to pair with 3, A with T with A, and so on. So between the bases, there will be hydrogen bonds, right? So adenine, two hydrogen bonds, thymine, 2 hydrogen bonds, guanine, cytosine, 3 hydrogen bonds, okay? So the pairing of the bases here, we call it as complementary. Complementary. Kita tak guna istilah same, same DNA sequence salah. The two DNA strand has the same DNA sequence is wrong. Sepatutnya the two DNA strand has complementary DNA sequence. The base sequence are complementary to each other. Okay. So complement. So yang ini saya cerita sikit, saya lupa. Kalau dalam satu DNA molecule, okay, so the basis is 100%. Kalau dalam satu strand ni kita ada adenine 50%, sorry bukan 25 lah. Thymine also 25%. Kena ada 25 adenine to pair with 25 thymines. Sama juga kalau kat sini ada let's say lah another DNA sequence, kalau kat sini ada 30 guanine, kena pair dengan 30 cytosine. That is rough, that's what it says here. The percentage of adenine and thymine are roughly equal and goes the same for guanine and also cytosine. Okay. Okay, so the second subtopic is models of DNA replication. Okay. Models of DNA replication. DNA replication is the process by which a double-stranded DNA molecule is copied to produce two identical DNA molecules. So there are three alternatives model of DNA replication. So the conservative model, semi-conservative model, and dispersive model. Okay. So basically, the DNA replication is where DNA is replicated from the parental cell to the daughter cells, from parental DNA to become two daughter DNA molecule. So how does ini? These are hypotheses or theory that were proposed to suggest how DNA replication occur. These are theories, models to suggest on how DNA replication is supposed to occur. Of these three, we have to know which one is the correct model that DNA replication occurs. Does the DNA replication occurs according to the conservative model, or does the DNA replication occurs according to the semi-conservative model, or does the DNA replication occurs according to the dispersive model? Which one? So for each of the model, you have to understand how the rules for each of the models. So they state apa. Okay, so this one, conservative model, semi, and also dispersive. Sekejap kelas. Okay, I guess we have to stop our session, our lecture for now, because it's already 9:50. So make sure before our lesson next time, which is tomorrow, you looked at what we are going to cover for tomorrow. Bacalah pasal konservatif, semi-dispersive, and all that, and all the possible topic that we are going to cover for 2 hours tomorrow. Bacalah. Supaya awak faham bila kita tengok topik tersebut esok. Okay. So we stop for now. Do you have any question? Ada soalan?
Tak ada.
Ada ya?